Can you hear me?
Yes, sir.
Okay, the first segment was over, so now it's, as I told you that after 10 minutes it will end and then I'll take you. So we have 4 participants here. Rest of the scholars, I think they know that they must join it again. Okay, they are joining. Sayyid Awais Shah is back. And rest of the scholars will also join. I have intimated you before it ends. Okay, let them join as well, rest of your friends. Okay, so that is called recombinant DNA technology, right? Now let's see. Recombinant DNA technology, there are two basic procedures: the basic procedure of recombinant DNA technology, and the second is application of recombinant DNA technology. First is the procedure that how we can obtain a piece of base pairs, a sequence of base pairs, a segment of DNA of our own desired. Why we talked about the base pairs and their sequence, specific sequence of base pairs, and we have talked about palindromic, that it must be palindromic, right? And all of you are now aware of this word. Am I right?
Yes, sir.
For to all of you.
Yes, sir.
Okay.
Yes,
So that's great. That's great. So it must be a sequence which must be similar from all the directions, from left side and from right side, like AGA, adenine, guanine, and again adenine, right? So we can call it AGA from either side, right? So this kind of sequence is known as palindromic sequence. So it must be palindromic in nature, and the sequence of base pairs matters. Why? Because a specific sequence of the base pairs will produce a specific type of protein which will be specific in nature and terms of function, right? So that's why it is very much important to get a specific sequence, and we must be aware of the sequence, that this type of sequence of base pair will generate exactly a specific type of copy of proteins. So we first of all, we must have education about, we must be educated about the sequence of the base pairs, and every type of base pairs and that type of sequence which produce protein. Like we have a type of base pairs, a sequence of base pairs which produce insulin, and then we collect insulin and we use it for some specific all of you are requested to please mute your mics. So what we said, that before that we must be aware of all the sequence, that this specific sequence will produce such type of proteins, and then every proteins play its role in different types of functions, right? So the first thing is to uh, get a segment of DNA which is required, which will produce a type of protein which is required to us. And then what is the application of recombinant DNA technology? We will talk about and this is, I think, the very last component of your syllabus. application of recombinant DNA technology, and we will talk about in detail. Okay, recombinant DNA technology, it begins with, as we have discussed earlier, with the restriction endonuclease enzymes. And what that enzymes do, it actually uh, select a gene of interest. A gene of interest or target gene mean that desired segment which encodes some specific type of information in the form of a protein, right? So that we can call it gene of interest, target gene, desired segment, specific sequence of base pairs, etc. And that target gene is then inserted into plasmid or phage. Now that gene which has been transferred to another phage or plasmid, that plasmid or phage would be called as vector, because what that would do, that will replicate that type of that recombinant section or complementary DNA. The replicon is then introduced into host cell to be cloned and either express the protein or not, right? So after that, we get the recombinant DNA segment, that particular piece, then it will be introduced or transformed into a host cell, a living cell, wherein they will start replication, and we can have large number of what we intend to get, the cloned copies, right? The cloned replicant is referred to as recombinant DNA, and the procedure, as you are well aware of it, it is known as DNA technology. Can anyone of you please tell me about the vector of malaria? Which organisms play a role in as a vector of malaria?
Anopheles mosquito, sir.
That's great.
Female
Anopheles.
Anopheles.
Okay. Anopheles is female mosquito and Culex is male.
Yes, sir.
It means that you know what vector is. All of you are aware of this term.
Yes, sir.
Okay,
Yes, sir.
Sir,
okay, that's great. Cloning, you know this term. Cloning mean to get the identical copy, right? As we have the very first word which we have heard is very much familiar with a doll. You know doll, dolly,
Yes, sir.
because that was cloned, and this
Sheep.
cloning term is strictly, familiar and attached, affiliated to that specific term. So we are well aware of cloning, right? To get the exact copy of anything, of an organisms or genetic segment. Okay, now we will talk about the steps of recombinant DNA. What are the steps? These are six in numbers: isolating vector and target gene, that which gene is required to cut that gene at specific positions or localities, and to get that, and to get another copy of that type, because we will have to cut another DNA. And I have told you earlier that we can get a sequence of base pair from animals, and we can ligate it with that uh, uh, exact pair of that base pairs of plant, right? So we can get a sequence of base pairs from animals, and we can ligate that base pair sequence with the plant. We can get it from bacteria, and we can ligate it with fungi. So this is, I think, very much interesting, that we can get that sequence from any of the organisms, and we can ligate it to that of the another sequence of base pairs of any kind of the organisms, right? So the first thing is to isolate vector and target genes. Then after selecting or isolating, what we do, we use restriction endonuclease enzymes for cutting or cleavage at specific positions and palindromic structure, right? It must be palindromic. And then what we do when we get the exact copies of target and vector genes, we ligate them by joining their ends and making them make bonds with each other, as thymine will bond with which specific base pair.
Adenine.
That's great.
Guanine.
And,
Oi.
who was this, Atifa or Mariam?
Cytosine
to
is not the
cytosine,
guanine,
adenine to
sir. Cytosine.
Yes, guanine will bond with cytosine,
Guanine.
or cytosine will bond with guanine, right? So adenine will bond with thymine, and if there is
Thymine.
thymine, it will bond with, it will, you know, it will attached with adenine. So
Double
thymine
bond.
and, yes, exactly, adenine and thymine and guanine and cytosine,
Cytosine.
right? So this will join, you know, after cutting, we will make a situation by using ligase enzyme to ligate them, and after ligating them, we will get a complementary deoxyribose nucleic acid. We will get a segment which is required to us, and after getting that complementary segment, what we will do, we will transform it. Where we will transform it?
So to the culture medium, sir.
To a host cell. That's great. To a host cell, we will transform it where?
Mostly bacteriophage is used for that. Sir.
Yes, exactly. So we will transform them into host cells where they will clone, where they will replicate that DNA, and we will be able to get the clone copies of what is, of what was required to us. And after cloning and after multiplication of that complementary segment, we can collect them, right? We will screen them and then we will select, because it is, you know, one of the very difficult and important thing to identify that clone copy, right? There we will get a numbers of DNAs, you know, the cell have its own replications and replicant DNAs. So we will first of all have to identify the cloned copies, and then we will collect them, because these were the segments which will produce specific types of proteins, and all those proteins which is required must have a sequence of base pairs which we have modified, which we have engineered. So if we will get the exact clone copy which has been engineered by us, so we will be able to get the specific protein which was required to us. If we get the wrong copy of any segment, if we get the wrong DNA, so we would not be able to get the specific proteins which was required to us. So selecting or screening is a very important step, and all these steps, these are the steps which we can talk about. It's not that it's not briefly elucidated, but it's only a general view. We can see isolation and purification, right? So both vectors and target DNA, they can be prepared, right? By there are lots of methods which can be used to get these target DNA, and they can be used to synthesize in vitro. Okay, then cleavage of DNA at particular sequence, and this is the most important step, because if we missed it, we may get a wrong sequence of base pairs, and if we get a wrong sequence of base pairs, we would not be able to get what we wanted. We would not be able to obtain the desired protein, because preparation or production of protein totally based on the sequence of the base pairs. So this is one of the important step where restriction endonuclease play an important role in specification of the sites where cleavage must takes place. Where cleavage must takes place would mean that there must be a sequence of base pairs which is required for the production of a required protein. Then if we get all those segments and we cleaves the DNA segments at certain positions, and at positions where we can get or the base pairs of our own choice, then what we do, we use ligase enzyme, DNA ligase, to ligate them and get a segment, a complementary segment. Fourth step is introduction of recombinant DNA into compatible host cell, where it will replicates itself and we can get them. Okay, replication and expression of recombinant DNA in host cell. Cloning vectors allow insert DNA. Insert is that DNA which has been engineered to be replicated, in some cases expressed in host cell, right? So the ability to clone and express DNA efficiently depends on the choice of appropriate vector and host. That is why we studied earlier that we will have to first choose the vector and target gene, because there may be some difference and there obviously are difference in the replications of enzymes in terms of vectors and host. If we use some vectors or host which take lots of times and lots of equipments are required to study them at different levels of these replications, it may be difficult for us to identify all these steps and then to identify the cloned copy of DNA. So for getting it easier, we have to select some vectors and host cells which is easily manageable, and from where we can easily obtain those recombinant DNAs, those segment which has been engineered by us, right? So in vectors we will see there it is mentioned even in these slides. We will see there are bacteriophages, yeast, viruses, different kinds of viruses. We will discuss all those types which are being used in these processes in detail. Okay, then identification of host cell that contain recombinant DNA of interest. Vector usually contains some genetic markers or genes that allows the selection of host cell that have taken up foreign DNA. And as we discussed earlier, that it's one of the most significant step or important step to identify that copy which has been cloned and which is required to us. So there are markers which are known as genetic markers or genes, which are used in identification of that particular type of DNA fragments which have been cloned. This is
Just a
DNA
gene type.
DNA cloning in a who is talking? What you said?
सर जेनेटिकल मार्कर्स इसके बारे में सर थोड़ा सा अगर क्लियर करें.
They have a specific type of genes and sequence which we can use to identify and to distinguish the DNA which has been replicated by the host cell, like their own DNAs, and DNA which are foreign. So there is a difference of the sequence of DNAs which we can obtain, and this is one of the component of the syllabus which we will talk about in detail later. We will now read it, and you will see the diagrams, so there it will become clear to all of you. Okay. Okay, now look at this. DNA cloning is a plasmid vector permits amplification of a DNA fragment. This is the diagram. it is like very much easy to understand it, right? There are and marker genes are those genes which help us and identification of those complementary DNA fragments. It actually broader them, it colored them, and you will see also in the slide is. So actually the plasmid or vector permits amplification on of a DNA fragments, right? Amplification mean that it make them, it allows them to replicate using machinery of the host cell. And you know why germs are being killed by the lymphocytes of our body? Who will answer it?
So lymphocytes are defensive system है body का sir, हमारा
That's
immune
really
system
great.
का.
Actually, this is the army of our
It
body.
identify the antibodies, antigens, sir, and produce antibodies against it.
That's great. It actually, what it do, it identifies all those foreign bodies. We can say foreign bodies. Germs are actually the foreign bodies which are identified by some of the elements of our body, that these are foreign bodies. These are not the bodies of our which is required to the body and which can be assimilated in the blood. These are some foreign elements which may create problem for the body. They identifies them and then they kill them, right? So this is how
Yes, sir.
marker genes do. They identifies those complementary DNAs. Did you understand now? They
Yes,
identifies them, and the mechanisms, we will talk about the mechanisms, that which is the mechanisms, what is the physicochemical structure which helps us an identification of those complementary segments, right? Okay, now how to get a target gene? The first is genomic DNA. Then genomic DNA mean that we are going to unwind it, then we are going to cleave it, and then we are going to get a sequence of base pairs which is required to us. Then artificial synthesis, as I told you, that we do it in a test, we can do it in a test tube, right? And then we transform it. Then PCR amplification, as we discussed earlier, and then reverse transcriptase PCR, right? RT, we will talk about it as well. First of all, polymerase chain reaction. What is polymerase chain reaction? It is a technique, and it has revolutionized recombinant DNA technology. What does revolutionized mean?
सर ये बड़ा चेंज है, इनकलाब लाया है इसने सर पीसीआर
That's
मशीन
good.
में.
It has actually aggrandized, it has culminated this technology. It has put impetus, or it has strengthened this technology, right? It is now strengthened. With this technology we can obtain recombinant DNA easily. So polymerase chain reaction has revolutionized, mean that it has given strength to this technology. It can amplify DNA from as little material as a single cell. Why it has revolutionized? Now it's the supportive statement. Look, as little material as single cell, and from every old tissue, such as that isolated from Egyptian mummies, and a frozen mammoth and insect trapped in ancient amber. Okay, now, as I told you, that from any source, we can get a segment, and to the and another segment of any source we can ligate both of them, right? And it is due to this PCR. It is due to this polymerase chain reaction. Okay, method is used to amplify DNA sequence. This is one of the diagram you can see in detail. The polymerase chain reaction can quickly clone a small sample of DNA in a test tube. Using this technique, we can get a cloned DNA very quickly and easily. These are the pictures you can see. Okay, RT-PCR mean reverse transcription polymerase chain reaction is a variant of polymerase chain reaction. In RT-PCR, however, an RNA strand is first reverse transcribed into its DNA complement or complementary DNA using the enzyme reverse transcriptase, and the resulting complementary DNA is amplified using traditional template RNA and then complementary DNA. So using this technique, anything is just possible. We can change the sequence of RNA, transforming it to complementary DNA. We can change the sequence of DNA, converting it to RNA. We can engineer DNA using the base pairs of, or altering or modifying the base pairs sequence of RNA. We can obtain RNA using or altering or modifying the base pairs of DNA. So this is very much, it's a huge development of this era. So we can manufacture things which is of genetic nature of our own well. And this is how they said that even from mummies and moths and dead moths and from anything we can get a sequence and we can utilize them, making the complementary DNA of our own choice. And this is not something like common or little, right? Now, vectors are cloning vehicles. Cloning vectors can be, what are the cloning vectors? As we discussed earlier, that there are viruses and there are types of viruses which we will discuss later in detail. Cloning vectors can be plasmids, it can be bacteriophages, it can be viruses, or even small artificial chromosomes. Most vectors contain sequence that allow them to replicate autonomously within a compatible host cell, whereas a minority carry sequence that facilitate integration into the host genomes, right? All cloning vectors have in common at least one unique cloning site, a sequence that can be cut by a restriction endonuclease to allow site-specific insertion of foreign DNA. The most useful vectors have several restriction sites grouped together in a multiple cloning sites, MCS, multiple cloning sites, called polylinker. There are unique cloning sites, right, which can be cut in specific positions. Those sites which can be cut using restriction endonuclease, and there are vectors which are having multiple such types of cloning sites. So those are known as polylinkers. This is the diagram. Okay, now types of vector as we discussed earlier: plasmid vectors, bacteriophage vectors, virus vectors, shuttle vectors can replicate in either prokaryotic or eukaryotics. Such type of vectors are known as shuttle and yeast. So in this in your syllabus, we will talk about bacteriophage, we will talk about yeast, and then we will talk about certain types of viruses which we can use as a vector. The first one is plasmid vector. Plasmid are circular double-stranded DNA, right? Okay, after 10 minutes, this session is also going to end. So what should we do? should we end it, or should I take you in another section?
Sir, it is sufficient for today, sir.
What rest of the scholars say?
Yes, sir. It's
All of
sufficient,
you,
sir.
all of you agrees?
Yes, sir. Yes, sir.
Okay, so let me tell you about plasmid vector, and then we will end, right?
Okay, thank you,
We'll
sir.
end this lecture. And before ending, I will also give you time to ask if you have something in mind, and we will discuss things together, right? Okay, so plasmids are circular double-stranded DNA molecules that are separate from a cell's chromosomal DNA. These extra chromosomal DNAs, which occurs naturally in bacteria and in lower eukaryotic cells like yeast, exist in a parasitic or symbiotic relationship with their host cell. Here is a bacterium. This is plasmid. You can see bacterial chromosomes and the plasmid. This is the structure of the plasmid as we discussed here. These are circular double-stranded DNA. So this golden circular DNA you can see at the right corner. This is actually the plasmids. Plasmid can replicate autonomously within a host, and they frequently carry genes conferring resistance to antibiotics such as tetracycline, ampicillin, and kanamycin. The express of these marker genes can be used to distinguish between host cell and that carry the vectors and those do not, right? So these are the vectors which we will tetracycline, ampicillin, these can be used as marker genes, and we will talk about their physiology and physicochemical structure and how they identifies between the host cell and vector cells. And next lecture, inshallah, in the coming slides, it is evident, and we can see that which are the factors involved in identification. As I told you that there are lymphocytes which identifies germs, and after knowing this mechanism, you would be able to understand even that mechanisms, that how they identifies the foreign bodies. You know, what we eat is a foreign body. What we take into our stomach or into our body, that is a foreign body. So then they identifies that whether this is good for the body or not. This identification and what is mechanism of this identification, we will talk about that in detail, inshallah. So what do you think? Was this lecture enough for today?
Sir, it is enough, and it was an excellent lecture today, sir. Very informative lecture, sir.
That's really great. So you might have learned a lot from this lecture, and if you have
Yes, sir.
anything to say, you can tell me. But before going into the detail, let me ask you a question. Can any one of you summarize what we read today in a summarized
Sir,
form? A
yes,
brief
sir, we talked
summary.
about.
Okay, that's great. Go ahead.
Sir, we talk about the recombinant DNA technology, how we get the recombinant DNA endonuclease enzyme that is used, and next step we ligate it, and we have a medium for the expression system that is bacteria most probably, and that is used to clone PCR. Also, we discuss in the MET. Sir, we can produce the gene of interest and for the use in the transgenic plants, in transgenic animals, in recombinant DNA through PCR machines, various recombinant DNA technology techniques. So that was the overall summary of the class, sir.
That's great. That's great. It was really great. Any one of you next?
Sir, today we learned something about recombinant DNA technology and its basic technology, two types, and the medias that are used for culturing the DNA. And then we learned that how recombinant technology starts by having a target gene, then a vector comes to take that into the host cell, and how cloning occurs. And then we learned about 6 steps of recombinant DNA technology, starting from isolation of the gene, then cutting, then ligating it, then transforming it, and after cloning, then selection of the screening of it, which was the most difficult step. And these were the 6 basic steps that we
That's
read today about it.
Excellent, excellent, very good. Next, please. That was really a good explanation from both of you. What about the rest of the scholars? Would you please play your role, or will you participate and telling us how can you summarize it? Like both of them, they summarize it in a very beautiful and excellent way, and there were variability of their summarization, and we learned from both of them, you know, in a few
Thank you, sir.
seconds, I think. So this was a very good explanation from both of you. Can you please, any one of you, anyone else, in your own words, like them? It's okay. What about the fast? How is it going?
It's good, sir.
Fast is very fast. Sir.
That's great. It's going fast, huh? And the weather is quite pleasant, so we
Very
can.
fast.
It's you know, really manageable.
Sir, yesterday you talk about the visit to Shangri district. Most welcome, sir, in Ramadan as well as after Ramadan, sir. When you wish, sir, along with Fidasim and other
And
all colleagues of my PhD class, sir.
inshallah, we will visit, and you will offer us a visit actually to all of your colleagues.
Sir, yesterday the line was cut up, sir. That was the main
Oh.
problem, sir.
Okay, it's okay. So I thought you were not willing because you know,
No, sir, I'm willing. It will be, sir, it will be an honor for me, sir, if you visit to my district, sir.
so nice of you. I was just, you know, kidding you. We know you are always there, and the first person who I saw whenever I open meeting. So what I saw
Thank you,
is, you
sir.
know, written Amir Sultan. So your name is
Thank you, sir.
Sultan, and you must also be a Sultan by heart. Huh?
Thank you, sir. Thank you, sir.
We were kidding you yesterday. I can understand these days there is a big problem of signals drop unexpectedly, and you know, when signals drop, then we cannot do anything until and unless the signals are restored.
Yes, sir.
Okay,
Yes, sir.
have a nice time, and wish you all good luck, एंड आपकी मुझे भी दुआओं में याद रखे और अपना भी बहुत सारा ख्याल रखे, ठीक है? टेक केयर ऑफ योरसेल्फ.
Thank you, sir. Jazakallah.
थैंक यू.
Thank you, sir.
अल्लाह हाफिज.
Thank you. Allah Hafiz.